everyone, and welcome to another video by BioTeach, this time focusing on the subject of immunity. I had some requests from Instagram followers to create something on this, and as it's such a large topic with some complex areas that you need to understand in quite a bit of depth, I've broken it down into a few sections. This first video will be looking at phagocytosis, and then I'll post videos on B cell and T cell action separately. This particular video is suitable for A-level students and BTEC students who perhaps do physiology units or units which look at disease and infection. The first thing we need to address are some key terms. I first want to talk about what is meant by an antigen and what types of molecules can act as antigens, and then I'll move on to how an immune system response is launched, and finally look at the step-by-step process of phagocytosis. I've also got some exam questions at the end that can help you practice your new learning. So let's first talk about what an antigen is. An antigen is defined as any part of an organism, which is often a protein on the surface of a cell, that is recognised as foreign by our immune system. The next question on there is, what can our immune system identify by their antigens? So things like pathogens, cells from other organisms of the same species, cancerous cells, and toxins can all be identified by our immune systems, and we can mount an appropriate response against them. Of course, sometimes our immune system does not recognise certain antigens or recognises them too late, by which time we become ill. We'll talk about that a little bit later. In this video, I want to identify the non-specific mode of immunity. By non-specific, we mean that it's a general response to antigens that enter our body. And this non-specific response is carried out by phagocytes in the process of phagocytosis. For your A-levels, you need to know about two types of phagocyte: neutrophils, which you can see on the diagram on the left, and macrophages, which you can see on the right. Neutrophils are cells that have a large multi-lobe nucleus, and they're produced in the bone marrow much like our other cells, and they circulate in the blood in large numbers. They are short-lived but increase rapidly in numbers to respond to an infection. They're actually named for their neutral staining cytoplasm and are the most abundant type of white blood cell. Some textbooks you read may quote that they constitute up to 3/4 of all white blood cells. They have this granular appearance under the microscope too, which is what's represented by those orange dots that you can see on the diagram. When they're activated, neutrophils become highly mobile and they can change shape. This is super important because that ability to change shape can allow them to travel along narrow capillaries to their destination where they need to be. They are some of the first cells to arrive at the site of an infection, and they're attracted by chemicals called cytokines, which are proteins that are involved in cell signaling. We'll talk about those a bit more when we look at B cell and T cell action. On the right, you can see a macrophage diagram. These are much larger than neutrophils, but are also manufactured in the bone marrow. In the blood, they circulate as monocytes, but then they settle in body organs like the lymph nodes and mature into macrophages. They play a part in coordinating the specific immune response because they ingest antigens, process them, and present them on the cell surface where they're recognised by T cells. In essence, it's that process known as phagocytosis. So we'll look at that in a second in diagrammatical format, and I'll give you the written version too. The first thing that we need to understand is really the role of phagocytes. Now, phagocytes have antibodies attached to the surface membranes, which will act as receptors. These will eventually be able to bind to the surface of other cells. And if we recognise an antigen as being self, as in our own antigens, then no response will occur. There is no need to mount an immune system response. But if it is a foreign organism with its foreign antigens, then it will bind to the surface receptors of the phagocyte and it will be recognised as non-self. It effectively triggers the phagocyte to begin the process of phagocytosis and start to destroy that particular antigen. So the first stage is when a phagocyte is attracted to bacteria by chemicals, or when a phagocyte recognises antigens on the bacteria as foreign. The phagocyte will begin the process of engulfment, which means it's taken up by endocytosis, as shown on the second diagram on your screen now. When engulfment is taking place, something known as a phagosome forms, and that's what you've got on the fourth diagram over there. Essentially, this is bacteria or antigens which are surrounded in a vesicle or a vacuole of its own. In this phagocyte, there'll be numerous lysosomes which contain powerful digestive enzymes. So a lysosome will effectively come along and fuse with the phagosome, and in this process it empties its destructive enzymes into the phagosome. This will allow the bacteria to become digested or hydrolyzed into its chemical constituents. Any indigestible material will be discharged from the phagocyte. There's this image over here of the whole process. You can see the steps of the bacterium being engulfed into the phagosome and the lysosome fusing with it. It's labeled as number 2 on there. And in this process, you'll have the hydrolyzing of the bacteria, as well as the exocytosis, which is labeled as number 3, of indigestible constituents. So it's the removal or the excretion of any materials that our cells are not able to use. I thought it'd be useful to have a couple of past paper questions within the video so you know the key terms to use. So the first one over here is asking about what an antigen is for 2 marks, and the second part is worth 4 marks, and it's asking how phagocytic white blood cells destroy bacteria. I'd like you to pause the video now and try to write down the answers to the questions and then play when you're ready. So the first part of the question, which asked you what an antigen is, for 2 marks, you need to state that it's a part of a molecule which stimulates an immune response. Note that you can say that antigens are proteins, or even name an antigen as well. Part B asks you to explain how phagocytic cells will destroy bacteria. So it's basically asking you to state the process of phagocytosis. For 4 marks, you had to say that the phagocyte is attracted to the bacteria by chemicals, or it recognizes the antigens as foreign. You needed to mention the engulfing and that the bacteria is in a vesicle or a phagosome. You should have mentioned that the lysosome will fuse with the vacuole and the bacteria is therefore hydrolyzed. Note that there are 5 possible marks here for a 4-mark question, so as long as you mentioned 4 of those points, you should get the full marks. Here's another question, this time for 3 marks. Describe how bacteria are destroyed by phagocytes. Please pause the video now so you can write down your answer. As this is for less marks than the previous question, you don't need to mention all of the previous points. Some of the important ones that I would mention are to talk about the engulfing. I would also talk about the lysosome fusing or emptying its contents to digest or hydrolyze the bacteria. Please note that for the last point, the mark schemes do not accept you saying break down or destroy. You have to use words like hydrolyze or digest instead. So hopefully that's nice and easy to understand. 3 points for 3 marks. Finally, there's a 6-marker question on phagocytosis. It says, describe and explain how phagocytes are recognized and how they destroy pathogens. So in this particular one, we're saying what happens and how it happens. So please pause the video now to give this question a go. So for 6 marks, you need to go into a little bit more detail. We know that we should mention words like antigen and perhaps maybe even say that they're foreign. I would mention engulfing by endocytosis. You'll notice that word invagination, that's just a posh word for folding in, so don't let that daunt you. Once the engulfing has taken place, we mention lysosome action, so we need to say that lysosomes contain harmful enzymes or strong enzymes that will digest the pathogen, and how harmless pathogens are removed, or harmless constituents of the pathogens are removed. And lastly, you should mention that the phagocyte displays antigenic components on the surface of its own cell, like a little flag, to start the immune response. Think of that cell waving a flag, letting other cells know that there's something foreign that the body needs fighting off. We call this an antigen-presenting cell now, or an APC. So we'll talk about that more in subsequent videos when we come to them, but that point is quite an important one to note. So please make sure you've got that written down as part of your notes that you might be making when you're watching this video. So I hope that was helpful, guys. I know it's a short video, but phagocytosis is a small part of immunity. The B cell and the T cell action are probably a little bit more detailed, so I'll be creating those videos very, very soon. Thank you so much for watching. If you've got any questions, then please post them below this video. Bye for now.